Hello everyone, this is bkoep from the Baker Lab. I'm back for another screencast. We're going to take a look at some more Foldit designs. Um, we're going to look at solutions from puzzle 1052. This is a 80 Residue Symmetric Trimer Design puzzle that closed about a week ago. Um, we saw some really cool things with this puzzle and talked a lot about them in our meetings, and I thought that it would be great for some Foldit players to get some feedback, and I think this is the best way to do it. So we'll start here. This is one of the top scoring solutions for that puzzle, and it's pretty typical of what we've been seeing here. These look like a very nice 3-helix bundles with a well-packed core, a good nice hydrophobic core, and then these bundles come together at a pretty nice interface, lots of hydrophobics. So we would consider that a very strong interface. These proteins would likely stick together very strongly. But I may have mentioned this before. These interfaces, in a sense, are a little bit too strong, in the sense that they're completely exposed hydrophobics. If you imagine just one of these monomer units, so take for example just this green one. You can imagine a full, almost 2 full faces of this monomer unit are completely hydrophobic. In fact, this helix right here is almost completely made up of hydrophobic residues. So I might not even expect this sequence to form a helix, and even if it did, you would expect this monomer to probably stick to just about anything else hydrophobic it could find in the cell. And the way to go about stopping this is a problem. This is something that you want to design against. A protein that sticks to everything is useless. We want a protein that sticks very specifically just to itself in this trimeric orientation. And the way to go about that, or one way to go about that, is to incorporate hydrogen bonds at the interface. And so that's what I want to talk with you guys about today. So I found some other solutions. These are not as high scoring solutions from this puzzle, but I pulled them out because they demonstrate some of the things that we want to focus on. So if I find the polar contacts here involving side chains, and these are not necessarily hydrogen bonds. Because I'm not sure about the orientations of all of these, but the distances between these polar atoms are close enough to be relevant for a hydrogen bond. So this is just an easy, quick and dirty way to kind of visualize where hydrogen bonds might exist in a protein. And this one you can see has a ton of them. A lot of these side chains are making very intricate hydrogen bond networks. And these are, in essence, these are the kinds of things that we would like to see, these buried polar residues between the interface that are typically more hydrophobic in the high-scoring solutions, but here are polar and form this unique hydrogen bond interaction that would kind of prevent, let's see, how should I say this? It would make this interface less strong against other hydrophobic targets. So this protein, this interface here would be less likely to stick to other things in the cell, but would still make a favorable interaction with this blue monomer over here. So these hydrogen bond networks, they don't really contribute strength to this binding interaction, but they contribute specificity, and that's really cool. Something like this right here, a network that contains residues from all 3 monomer units. That's really nice. Of course, the problem with this particular design is that there are a bunch of these polar residues in the core, and while they may form some hydrogen bonds, that's not what we want. In the monomer unit, we want strictly hydrophobic residues in the core. Um, when we think about a monomer folding, especially a small one like this, we don't really have to think so much about specific interactions. We just want strong interactions. It's very unlikely that this thing would be unlikely to fold up just because it makes interactions with another molecule. Um, so we'd really like to keep the internal residues of the monomer unit hydrophobic. Um, and again, most of the, or at least a lot of interface residues should also be hydrophobic, but we want um, some select polar hydrogen bonds in between these monomer units um, that will really confer some specificity between these units. Another problem here, um, this serine right here, it looks like it's kind of buried, so it doesn't have great access to solvent, and yet it doesn't make any hydrogen bonds, and that's not good. Uh, when you bury a polar atom, you really need to satisfy all of its hydrogen bonds, and this kind of goes back to that idea that in the unfolded state, this monomer, this serine here, would make a hydrogen bond with water, and in this folded state, it's not making the hydrogen bond, which means that the potential energy of this folded state is a little less favorable, and means that this whole thing is less likely to form when you have an unsatisfied buried polar atom right here. And we'll talk a little bit more about this maybe in a bit. But yeah, so these are the types of things we'd like to see. Some hydrogen bond networks. I think I have another one here that looked interesting. Again, this is not a great scoring solution, but it does exhibit some cool hydrogen bonds, cool hydrogen bond networks. Yeah, this one is pretty cool. So let's see. Let's look at. Yeah, wow. So if we look at these residues here, we have this arginine, a glutamate, see some asparagines, aspartates. So this is really cool. There's a tyrosine that's involved. This is, if you look at this network right here, it contains, it includes several residues across each interface. So there are a lot of inter-unit hydrogen bonds, and there are also a lot of hydrogen bonds backing this up. So when you have a hydrogen bond network like this, it's nice to have a lot of residues contributing to the hydrogen bond network, because it makes this network stronger when you have, for example, this tyrosine and this asparagine down here to kind of back up the positions of these residues. It really locks down those residues into this conformation and makes it very likely that these would form this hydrogen bond network. So this is a really cool network. It's pretty extensive. Again, other problems with this design, there are some polar residues in the core of the monomer. These helices don't look especially great. They're kind of bent and a little twisted. Again, there's not really much more of an interface between these monomers beyond these hydrogen bonds. So in this case, you have specificity possibly here, but there's no really hydrophobic interface that would lock these monomers together. So the interaction between them is weak, if specific, and that doesn't meet our goals. We want a strong interface that's also specific. Let's see. I do have, this is an example of something that one of our scientists in the lab has come up with. This is a hydrogen bond network that they designed, and a cutaway of a helical bundle here. And you can see that these asparagines here all line up very nicely and satisfy all of the possible hydrogen bonds between between these atoms. So while these polar residues are buried, their hydrogen bonds are satisfied, and these helices would be expected to come together in a very specific orientation. And of course, the other side chains are visible in this image, but there would still be plenty of hydrophobic residues between the interface and just this one or maybe two networks in the whole unit, in the whole multimer, that would keep these guys coming together specifically. Yeah, so this is cool. This is something that we'd really like to see more of. We're working on getting support for hydrogen bond networks into Foldit. I think that this is something Foldit players would be very good at. The way we're going about this in lab is I think it can be improved. I mean, I think that Foldit players might have an advantage over computers in the sense that you can more easily change conformation and design these hydrogen bond networks visually, whereas the computer is kind of just trying lots of different things combinatorially and stupidly. So I'm really looking forward to getting this hydrogen bond network stuff into Foldit, so that hopefully things like this or networks like this can contribute to the score, and we can start rewarding players for doing these really cool things. Let's see. I have some other solutions from this puzzle that I thought were really cool. These are shared with scientist solutions. Yeah, this is one that looks really cool. It's an interesting topology. There's a big error here, though, and it relates to what we were talking about before with buried polar atoms that do not make their hydrogen bonds. And they're difficult to see in this mode because we're in ribbon mode. I don't have all the backbone atoms visible. But if we show these as sticks, this strand right here, these residues that make up this last strand at the interface, they have all of these polar residues that are sticking out at the interface. So we have this carboxyl oxygen, this amide nitrogen, again over here, these atoms as well. Those are atoms that want to make hydrogen bonds, and the protein will not fold up in a way. It will not fold up into a shape where these atoms do not make hydrogen bonds. So this interface would be very unlikely to form. These residues really need to have hydrogen bond partners to have a stable structure. So when you have something like a sheet here that's butted up against a really hydrophobic interface on the edge, that's no good. We don't like that. So as cool as this looks, and as nice as some of the interactions look, and the kind of complementary surfaces, this is not very promising for us. Here's another one that's a little more reasonable. We have now the sheets are a little more solvent exposed on the edges. This one's definitely fine. Excuse me. This one on the inside a little bit. It's a little close, butting up to this other interface. But what's cool here is that this tyrosine and this glutamine are backing up some of those hydrogen bonds right here. So if we let's see if we find polar contacts, and then I'm gonna show the side chains. Let's see, can I do it for just this? Yeah, so here we go. So we're looking at this this green unit. Yeah, so you can see these carboxyl groups on the edge of the strand are satisfied. They're making hydrogen bonds with these side chains over here. So that's a really cool interaction. This is pretty cool. So we will probably be asking Foldit players to improve on this design. Um, Again, other things we notice with this, that the interfaces between the subunits are very hydrophobic again. There's this huge hydrophobic face right here that is not very specific. So it would be really cool if players could incorporate some hydrogen bonds in between these. Um, there are kind of a lot of methionines in here. We don't really like packing methionines too much. If I recall, I think there's a little bit of a void here. It looks a little empty. Yeah, there's kind of this empty space, some voids running through the middle of this protein, so it could be packed a little bit better. Um, but we do like this topology. We like the beta sheets on the outside. Really like these side chains backing up the backbone atoms here on this last strand. And the packing otherwise is looks pretty good between these guys. So this is a really cool shareware scientist that we liked. So you have another one here. Oh yeah, this one's really cool. So here we have some beta sheet pairings between the monomer units, and this is something you see in nature sometimes. It's a really cool way to kind of to contribute to some specificity, because what you'll see is that these sheets right here make hydrogen bonds across each other. Like, let's see, yeah. So as you guys will recall, beta sheets they're stabilized by these hydrogen bonds that form a kind of hydrogen bond ladder between strands. And so here you have the exact same thing, but since the strands are between different monomers, these strands belong to different monomers, these cross hydrogen bond ladder contacts really contribute to some specificity in a way that's pretty cool. We like to see things like this. This is actually overall a really interesting and cool topology. It has a surprisingly kind of compact core with just 80 residues. This is it's kind of impressive. Some of these loops are kind of long for ideal proteins, which is kind of unfortunate. And then this helix right here is almost completely hydrophobic, so it might not fold up. Again, these interfaces are mostly hydrophobic. Could use maybe another hydrogen bond network somewhere in here. Because remember, again, those polar residues on the surface increase solubility, which is also very important for the monomers. And then the last thing, there are also a lot of tryptophans here. I count 4 tryptophans in an 80 residue monomer, which is kind of a lot. We don't need that many tryptophans, and other residues I think would pack a little bit better. So yeah, this is actually another really cool topology that I think we might try to recycle and ask you guys to try to improve. It's really very cool looking. Really like this one. This is cool. Yeah, and there were some other ones that do similar strand pairing things. So here's one. So you have the same sort of thing where you make a interchain beta sheet using beta strands from 2 different monomers. And this looks pretty good for the most part. The edge strands are exposed to solvent, so they'll probably satisfy all of those hydrogen bonds we've been talking about. The problem here is that this monomer unit is really very spread out and not very compact. There's not really much of a core in this single monomer unit, so we would be very surprised for this thing to fold up and make these correct interfaces. one thing that's cool. It looks like there is a bit of a hydrogen bond network here, right, and these three serines in the middle of the interface. But aside from that, there's not really a whole lot to these interfaces right here. Wouldn't really expect this guy to fold up or form a strong binding interaction. So yeah, so there you have it. That's all for today, I guess. Look out for some hydrogen bond network support and Foldit in the near future. We'd really like to get this in because I think that's the main thing holding back Foldit designs these days, and it's also something I think Foldit players could really excel at. I'm really excited to see what you guys can do once we get these things contributing to the score. And my understanding is that this should be pretty fast too. So I know we keep throwing really slow filters at you guys, but hopefully this is one that should be a little faster and easier to run. So yes, great folding. Keep up the good work. We'll look forward to seeing more Foldit designs from our wonderful Foldit players. Thanks everyone.